Hi everyone, today we are going to do the experiment protein precipitation by organic solvent. Generally, the protein precipitation using the organic solvent can be done with the acetone. Here we have two Eppendorf tubes, one Eppendorf tube containing the distilled water which is acts as a control, and another Eppendorf tube contained the intracellular protein which was obtained by disrupting the bacterial cells using the chemical method. Now what we have to do, we have to take the protein present in the Eppendorf tube, then we have to transfer it into the 25 ml falcon tube. So the entire 1 ml we are taking through the micropipette and dropping into the falcon tube. So after that, for the precipitation, we have to add 8 ml of pre-chilled, which was kept in the 4 degrees centigrade in the refrigerator, the acetone. We have to add 8 ml into the protein sample. So the pre-chilled acetone, which will enhance the precipitation, that's why we are keeping it in the refrigerator. Now after adding the acetone 8 ml, what we have to do is we have to keep that acetone containing the protein mixture in the minus 20 degrees centigrade for 30 minutes for incubation to ensure the protein precipitation. So after adding the acetone, we have to mix it vigorously so that the acetone will be evenly mixed with the protein sample and it will initiate the protein precipitation. So after vigorous shaking, we have to keep that the acetone protein mixture inside the minus 20 degree centigrade deep freezer for precipitation. So after the precipitation, that means after the 30 minutes of incubation, we have to take that protein and we have to centrifuge it so that the precipitated protein will form a pellet. In opposite side, we have to keep the balancing Eppendorf tube containing distilled water. The centrifugation we have to do it in the 10,000 RPM for 10 minutes. Generally, the acetone precipitation, how it happens means the protein have the higher solubility in the water by sharing the hydrogen bonds with the polar amino acids. This acetone will disrupt that hydrogen bond forming between the water molecules and the protein and ensures that protein precipitation. Here you can see, after we have taken out the acetone precipitated protein, we have the pellet on the bottom and we have to keep some more time in the room temperature to ensure the precipitation. So once the precipitation is done, we have to discard the entire acetone and we have to resuspend the pellet in the distilled water. Now the protein pellet, which was precipitated using the acetone, was mixed with the distilled water and vigorously shaken. Then we will start the measuring the protein concentration using the UV spectrophotometer. So the UV spectrophotometer, we are measuring directly the protein concentration at 280 nanometer. First we have to blank it using the distilled water so that we will get a 0.00, that is the blank value, just a distilled water. Now if anything difference in the OD value at 280 nanometer, the concentration of the sample that we are keeping in the UV will show the reading. Now the protein that we redissolved in the water, we are taking in the Eppendorf tube inside the cuvette, and the transparent regions of the cuvette we have to wipe it out so that there must not be any disturbance while measuring the OD. Now we keep the measure means we can get the protein concentration, that is 0.085, showing the protein precipitation. Thank you.